Welcome back, colleagues. As researchers and clinicians, we literally spend our entire careers looking for the next big frontier in cellular health and tissue repair, right? Well, today, in this explainer, we are focusing our lens on exactly that by examining the foundational biology of exosomes. We're going to break down the exact mechanisms and payloads of these microscopic messengers, translating all this latest source data into actionable clinical insights for your practice. Let's jump right in.
Okay, Section 1, the nanoparticle network. Let's dive into this and look at the structural foundations.
So, by definition, exosomes are nanoparticles. But here's the crucial thing. They aren't foreign or synthetic entities at all. They are actually released by nearly all cells in the human body. And when the body universally utilizes a specific structure like this, it is totally fundamental to our biology. But what's really exciting, especially for dermatology, is that skin cells actually possess their very own highly unique kind of exosomes. This specialized, localized adaptation at the dermal level, it's exactly why we're seeing such a massive pivot toward exosome therapy for treating things like photoaging, severe dermatosis, and just general cellular aging. It means we aren't just dealing with a generic biological tool here. We actually have a highly specific key designed perfectly for the unique lock of human skin tissue.
Moving right along to Section 2, the exosome payload. Let's unpack the biochemical composition.
Okay, so when one of these nanoparticles is released, it doesn't leave empty-handed. It actually carries a highly specific, incredibly dense mixture. We're talking lipids, proteins, amino acids, peptides, growth factors, and genetic material, the whole shebang. For us as clinicians, this isn't just some basic structural envelope, right? It is a highly complex, biochemically active delivery system. Because they're carrying these precise genetic instructions and incredibly potent growth factors, they are fully equipped to fundamentally alter the cellular environment they interact with. I mean, they contain the exact building blocks required to execute really complex biological repair processes right at the cellular level. This effectively bypasses the need for the cells to synthesize these vital compounds from scratch when they are in a crisis. It's actually quite brilliant.
All right, Section 3, the regeneration messengers. Let's examine their actual mechanism of action.
So how do these loaded nanoparticles actually function in the tissue? Well, this foundational pathway operates in two distinct steps. Step 1 is cell-to-cell communication. Essentially, exosomes act as the physical bridge between the originating cell and the target cell. Then Step 2 is signal transmission. Once they're connected, they actively transmit signals derived from that dense payload of proteins and genetics we just talked about. They're essentially the active operators in the body's cellular network, constantly routing crucial information across the cellular divide to orchestrate repair. This basically ensures that the biochemical payload is delivered with absolute pinpoint accuracy to exactly where the tissue requires the intervention.
And this brilliantly illustrates the core concept. You can really think of them as messengers sending signals to unhealthy cells, triggering them to regenerate. As researchers, you know this targeted mechanism is basically the holy grail. Exosomes specifically seek out compromised tissue. The growth factors they carry act as a direct trigger, initiating the regeneration sequence exactly where it is failing, rather than just broadly and inefficiently flooding the whole system.
Which brings us to an absolutely crucial clinical distinction. Exosomes actually prompt the body's natural healing process. Now think about standard healing methods or traditional pharmacology for a second. We are so often forcing an extrinsic chemical pathway upon the tissue, right? But exosomes rely entirely on the body's intrinsic system for self-repair. They literally kickstart a natural healing cascade that the body already knows exactly how to execute. So we are working in tandem with human biology here, rather than trying to override it. By utilizing a pathway the body inherently recognizes, we bypass all that resistance and the complications we so often see with synthetic interventions. It clears a path for profoundly efficient regeneration.
So, knowing that skin cells have their own unique exosomes packed with these regeneration-triggering growth factors, I want to leave you with this final thought. How will harnessing these microscopic messengers fundamentally transform our approach to skin regeneration? Mastering the application of the body's own natural healing triggers could literally render our current standard of care obsolete. Thank you so much for joining me for this explainer, colleagues. Keep researching, and I definitely look forward to exploring the next big breakthrough with you soon.